Good morning. So anyway, I appreciate very active discussion. So 10 minutes behind. So anyway, next session is biotechnology and bioengineering and cardiovascular science. So chair is I am Tatsuya Shimizu from Tokyo Medical University and Professor Hill from UT Southwestern Medical Center. So this session includes 3 speakers, including me. So the first I'll introduce the Professor Adam J. Engler from the University of California, San Diego. So the title is Dynamic Biomaterials Improve Cardiovascular Disease in Dish Models. So please start.
All right. Well, thank you very much, Jay and Kichi, for the invitation to tell you a little bit about what we've been doing in San Diego over the past several years. I just have 2 really brief disclosures about technologies that we've developed over the years, and I just want to thank those in particular that did the vast majority of the work. Those that are highlighted in bold here really did the lion's share of the work since I've been out of the lab for so many years now. But it's been a long-standing pleasure to collaborate, especially with Kristen Baldwin, who's since left scripts for Columbia, as well as her former postdoc, who's a very talented new assistant professor, Valentina Losardo at University of Wisconsin-Madison. And so a lot of what I'll show you today has been really the fruits of that very nice collaboration. And of course, I want to thank NSF, NIH, and the Wu-Tsai Human Performance Alliance for all of our funding. I think it goes without saying, based on some of the conversations that we had yesterday, that everyone should appreciate that the cell is a 3-dimensional object that lives in a 3-dimensional matrix. Oftentimes when we're dealing with cells on tissue culture plastic, we sort of gloss over the fact that this is a 2-dimensional culture substrate. And so what I'll tell you about today is really some efforts that we have undertaken over the last decade to really push cells into more 3-dimensional and representative environments, or at least in 2-dimensional environments that provide specific extracellular cues to those cells. And what are those cues? Well, they include things like the topography that the cell is living in, so whether it's smooth and flat or fibrillar and 3-dimensional. The ligand concentration and composition, so are you talking about lots of collagen, what type of collagen, fibronectin or laminin, the shape that the cell takes on, especially in a 3-dimensional environment where the matrix can be very porous and have large holes or very small holes, and that dictates how the cell spreads out into its environment. And last but not least, then the difference between something that's relatively very rigid, like tissue culture plastic, and something that is unfortunately way too soft. So that is my one joke. But ultimately, we want to think about how matrix can impact disease, and we want to use some engineering approaches to really understand how we can change the environment and see how cells respond to that. So, of course, I think we can all appreciate that the extracellular matrix can remodel with disease. So here we're just showing an AFM atomic force microscopy profile of a rat myocardium that underwent an infarct, and everything downstream from this point where the arrow is was occluded and subsequently remodeled and became a stiffer, hard scar that you can see by trichrome imaging in the upper left. But the thing that, when thinking about these microenvironments that we're really good at is providing specific cues, some of the ones that I just mentioned, to iPSCs that have very unique signatures to them. And I think that we can all appreciate from the conversation yesterday again that the vast majority of our genomic DNA is non-coding, but we shouldn't necessarily then get rid of it owing to the fact that about 80% of it has some sort of biochemical activity. And I'd point out in particular for what we're interested in today that there are over 28,000 known long non-coding RNAs. And interestingly enough, no one knows if any of them are mechanically regulated. So we wanted to grab this problem and really delve into it with our engineering approaches. And one of the reasons why we want to use IPS as a model system for this and to do some more complicated disease modeling is the fact that a lot of the single nucleotide polymorphisms that exist in our population are found in non-coding regions. And they can be associated with significant risk where the only opportunity that you have to model that risk is in an IPS system, because these SNPs developed in animal models that are higher than mice and rats, typically in like a non-human primate. So this is a Manhattan plot if you're not familiar with it, and basically it's just pointing out the association between risk and the chromosomal address for it. And I'd like to draw your attention to this tallest peak here. This exists in a region of our genome called 9p21, and 9p21 is the second largest gene desert in our body. It's associated with about a twofold increased risk of early onset myocardial infarction, which is approximately equal to the amount of additional risk that you take from smoking. Moreover, this is a really prevalent set of SNPs that exist in our genome, whereas about 1 in every 4 of us is a homozygous carrier for these SNPs. I'd also mention that these SNPs exist in something that we call linkage disequilibrium, which is a fancy way of saying that if you have one of them, you tend to have multiple, if not all of them, in this one region. So again, in this great collaboration that we had with Valentina and now her newly formed lab at Wisconsin, we started looking at chromosome 9 at this one region and noted that, despite the fact there are a number of SNPs here, what it really provides is an opportunity for us to edit the genome and to pull out this one risk region. We can't use modern editing tools like CRISPR because that would come in and edit single variants. And like I said, because of linkage disequilibrium, you're generally going to have many, many of these SNPs, and so the likelihood that you can correct all those different variants is almost impossible to do. So, so we created our risk and non-risk patient populations, made iPSCs, and then corrected them via TALEN-mediated genome editing, and this just shows you in the bottom plots here that we can in fact make our iPSCs out of these edited cells. So what is the most important take-home specifically from the disease mechanism itself is that it seems like all of these cells, whether they have these risk SNPs or not, will express a significant amount of the long versions of a long non-coding RNA called ANRIL. And that differential splicing or alternative splicing leads to shortened versions of ANRIL that are preferentially expressed in patients that have these SNPs. So as you can see here, these short versions then are upregulated as cells are differentiating. So the iPSCs themselves express very little, they have very few differentially expressed genes. But when we look over the course of differentiating these cells down whatever vascular progeny you're interested in, we see a tremendous increase in the number of genes that are differentially regulated by this one allele. So our engineering approaches now sort of come into play in that we take these patient populations, we've edited them in steps A and B here, and now we create different engineered environments, whether we call it a heart attack in a dish or a microvascular network that we're interested in probing further. So to get you started on our cardiovascular efforts, one of the things that we noticed over the years is that we could take materials and engineer specific sort of smart functions into them. And what I mean by that is, we aren't born with a hard infarct scar. This develops over time. So if you were to give cells an environment that was, too rigid or relatively, soft, that wouldn't necessarily provide the right dynamic stimuli to them. So we could create these methacrylated hyaluronic acid hydrogels that cross-link slowly over time. So you can see sort of in multiple steps, we can go from something that is relatively myocardial-like to something that is more fibrotic-like. So if you take home nothing else, again, take home the fact that we can put these cells into these stiffer soft myocardial-like environments, and they will beat regularly. If we put these same cells that have the risk background into a cell culture where the environment has now been stiffened, we see a loss of that coordinated calcium flux. So interestingly enough, we can take our genome-edited cells and ask this question in a much more refined manner by saying in the risk knockout versus wild-type scenario where we're making an isogenic comparison, what happens? And it turns out that we see preferential loss of the connexin 43-based junctions here. So these gap junctions disappear in the risk wild type only when we've stiffened that environment. So in sort of a normal myocardial stiffness, they form very nicely. Now again, we think that this is due to differential expression of anryl, and so if you make this comparison, say in the short anryl isoforms, the normal wild-type populations have a relatively low expression of these, and then the risk patients have a higher expression of that. So I don't have enough time to tell you the full story that involves JNK and Krebs signaling, but suffice it to say that there are really important downstream effects that transcriptionally silence the connexin 43-mediated junctions. So we again wanted to go back to our engineering toolkit and say, well, if the junctions are the problem, is there a way that we can get around these junctions? And in some experiments that Giselle Gonzalez did in the lab, she said, well, maybe we could take conductive polymers and we could spin them into a system that makes it look like extracellular matrix. It's three-dimensional. The cells can intercalate into the matrix, and they can bind to it and begin to electrically signal, even at very low sort of subconfluent densities, because this material now is conductive. And so the data that I'm showing you in this plot on the right-hand side, you can just see the increase in electrical conductivity as measured by these substrates. So we can put down our cardiomyocytes again. These are just PSC-based myocytes, and just look at differential expression of the genes from the transcriptome here. And you can see very nicely some of the ontologies that come up are things like heart contraction, heart process, muscle and muscle contraction. So that made us feel really confident that if we put these subconfluent cell layers into the material, then that they could still electrically couple to each other. And that's in fact exactly what we see. As we're moving from left to right in each of these graphs, we're increasing the amount of electrically conductive polymers in the system. And so you can appreciate, hopefully, that the blue data shows the highest amplitude of calcium flux. It shows the most coordination, the quickest time to peak. And all of this can be done in a system that is subconfluent. And we can compare that then to our confluent systems, and we see the same sorts of conduction concentration mediated effects. So I just want to take a step back for a second and say, well, you know, if there are beneficial effects from engineering the environments around these cells for cardiomyocytes, what are the implications then in vascular progeny? So for the rest of the talk, I just want to tell you a few really quick vignettes of differentiating these cells from a mesoderm into vascular endothelial cells and vascular smooth muscle cells. So to think about the smooth muscle story for a second, and this is something that literally will come out next week in PNAS with Jamie Maynor as the lead author here, we noticed that a lot of these individual patient lines and then their clones themselves would exhibit a tremendous amount of variation in terms of both size and morphology, and that led us to wonder, well, are there underlying differences then in terms of the heterogeneity of their responses to their microenvironments? So we took a step back and said, okay, well, let's just again look at the transcriptional profile of these cells and maybe the integrins that they're using to adhere to their environment. And interestingly enough, if we look at this red column here, these are all patients that are derived from risk backgrounds, or cells that are derived from risk backgrounds. And you can see very nicely that there's a differential clustering between integrins that are selectively turned off and those that are turned on. And that led us to wonder, well, maybe there's something a bit deeper in terms of what regulates the heterogeneity. So again, going back to our engineering toolkit, we can not just make biomaterial systems, but we can make microphysiological systems. So here is a microfluidic system where we can put our cells into this chip and apply very specific shear stresses to those cells. And when seeing under which conditions would cause these cells to detach from their microenvironments, across different patients, we saw different results. So if you were to look at risk patient 2, these cells very nicely separated from each other and had different phenotypes, meaning that the more weakly adherent cells were smaller and looked more synthetic-like versus contractile. But that wasn't necessarily the case with the first risk patient that we dealt with in all of its clones. So it just happens that different shear stresses seem to sort these populations, and so we could find other shear stresses, so 90 dynes per centimeter squared versus 30, that then sorted these patient populations. Now, there were knock-on consequences to this in terms of the phenotypes that these sorted populations showed. Whether we're talking about how contractile they are, so if you look at weakly adherent cells versus strongly adherent cells, the amount of traction stress or the energy that they can impart when they're contracting is different. Their alpha SMA concentrations are different. Integrin expressions are different. So we thought that there was something really fundamental about the heterogeneity in this system. And what seems to underlie it again is variable penetrance of the expression of ANRL. So what I'm showing you here is just a diagram of all the different ANRL differential splicings. And I want to focus in on A2, 3, 11, and 12 because that's what we found from our RNA sequencing. So we could come back and look at the expression of ANRL again in the short isoforms here. And again, you see our weakly adherent cells are very nicely overexpressing the amount of ANRL here, where there's no difference in the expression then of these long versions of ANRL. So then we could go back and we could engineer into our system, and again with Valentina's help we were able to create sort of an add back, if you will. So we knock out ANRL expression, we then add it back with a viral vector, and we can again rescue the fact that weakly adherent cells in the presence of this ANRL differentially spliced short version then is non-contractile and very synthetic. So in the last 2 minutes, I want to just diverge one final time and say, well, if we see differences in the presence of vascular smooth muscle cells, do we see any changes then in vascular endothelial cells? And so, not surprisingly, the answer is yes. But first we wanted to just say, okay, again, our cells, independent of whether or not they are from a risk background or not, in the absence of any sort of mechanical stimuli, their transcriptional profiles are relatively similar as shown in these PCA plots. So we put these cells into a little bioreactor that recreates a patent vessel. And so this blood vessel in a dish we can show has very nice junctions, and it's patent as shown by our confocal microscopy here. So you can see that they are hollow tubes. And then we can perfuse them. And interestingly enough, when you perfuse them at very low wall shear stresses, so something that would be subphysiological, we don't see any sort of rearrangement of these cells, whether or not the risk is present or not. So in the knockout case and also in the risk wild type. But if we expose these cells to very high shear stresses, now we do see rearrangements in our risk knockout cell lines, but I want to draw your attention to very large holes that actually develop in the tissue-engineered blood vessel that we have for the risk wild-type patients. And these holes then preferentially allow fluid to exchange with its surrounding environment, so the permeability of these cells go up, of these tissue-engineered blood vessels. So somewhere around a supraphysiological or pathological shear stress, now we're seeing an upregulation or an elevated amount of permeability. So again, our transcriptional profiles now diverge in the presence then of this high wall shear stress. And that can lead to the upregulation of a series of genes that are associated with coronary artery disease. So if you look at gene profiles like cardiogram and those sorts of efforts, we see sort of a flip-flop between sort of low expression of all of these cardiovascular genes, and then the CAD genes will be upregulated in these wild-type cells when under the influence of that acutely high shear stress. Now, this also can be confirmed in patients. So when we're looking at ascending aortas, and we look at the aneurysms that can result in this disease pathology, the patients that we can take these little rings from that are undergoing root resection, then we can fix and stain them. We see a tremendous amount of proliferation in the aneurysmal regions. But if we just compare sort of normal fluid flow regions as identified by 4D flow MRI, and we compare that then to the aneurysmal regions of patients that do or do not have these SNPs, you can again see elevated wall thicknesses with these patients. So we think this is really nice confirmation that the smooth muscle cells in this layer can become hyperproliferative. We can also lose then a very nice endothelial layer that exists on the luminal surfaces of these cells, of these aneurysmal regions of our aortas. And so that again takes us back to this final concept that if we're using engineering tools to interrogate our microenvironments in iPS lines where we can only mimic disease using those sorts of mechanisms as opposed to animal models, that perhaps these tools can be really useful. So if I've convinced you of nothing else, hopefully it's that we can use these tools to sort of reverse engineer the niche that are present with these cells, whether it's stiffening the microenvironment with biomaterials or changing the fluid flow shear stress profiles to sensitize endothelial progeny in 2 and 3 dimensions. Hopefully you can take those home and apply those tools to your own investigations with iPSCs. So again, I just want to thank you for your attention today. I'm happy to take any questions. And again, thank you so much to the organizers for the invitation. Thanks.
Well, thank you for nice talk for Professor Engler. So please start.
Adam, great talk. Just to be sure, so this 9p21 region is associated with increased shear stress on the arterial wall. It has nothing to do with high cholesterol or inflammatory conditions. That's the mechanism for coronary artery disease. So if that's the case, why is it specific to coronary vessels, not other vessels in the body?
It's a great question. We think that it's based on just the differences in the sort of absolute shear stresses that these cells are going to see. So in capillaries, the shear stresses are going to be orders of magnitude lower than what we would see in these disturbed flow regions in, say, the ascending aorta or in the coronary artery. So I think that it's just sort of the magnitude of the shear stress that they're feeling.
So in that case, you would expect higher rate of stroke in those patients too, right?
Mm-hmm.
But we don't see that, right?
Yes, which is what keeps us in business trying to study this. I apologize for the glib answer, but you're right in saying that there should, we would expect to see correlations with stroke as well, though those aren't nearly as strong as the cardiovascular correlations that we get in this locus.
Okay. And just to be sure, you have to be homozygous for your risk to be increased, right?
We were only looking at homozygous,
So
yes. There is a slight elevation in risk based on those GWAS studies that I cited for heterozygous carriers, but because the stories are a bit more muddled in that, we just focused on heterozygous risk and non-risk, and we left everything in the middle alone.
Great. Thank you.
Okay, time is limited. Simple question, one by one. Yeah.
Hi, really enjoyed that talk. Can I just ask you about, you made a comment that the phenotypic variability that you were seeing was caused by the variation in short forms of ANRL, is that right?
Mm-hmm.
But what you showed was an association, or did I miss that? Did you actually modify the expression of ANRL?
Yes, so in the add-back experiments, what we did was we knocked out the locus, and then we virally added back just A11 and A12, which are the short isoforms of ANRL. So I apologize for glossing over that. That's all going to be in our PNS paper next week, and I'm happy to send it to you.
And my other question was, do you see the similar phenotypic variability in other smooth muscle lineages, such as neural crest-derived ones? You just looked at mesodome, I think.
Yeah, no, we've only used Chad Cowan's protocol to make our smooth muscle, so we haven't looked at that yet.
Also really enjoyed your presentation. I was wondering, in the endothelial cells, smooth muscle cells between different vessel beds differ substantially in respect to expression profiles. So I wonder, with your artificially engineered vessels, well, which type of vessel bed are you reconstructing? And also, is it possible that such differences might also account for some reactions in coronary vessels and pulmonary vessels, and so on? So primarily, I mean, what type of vessel bed does your construct reflect?
Yeah. So to be clear, when we're doing all of our vascular smooth muscle cell work, those are still monolayers, as are in the tissue-engineered blood vessel. We just have a lumen with a coating of endothelium. So our goal is really to provide or to develop a layered system where you have layers that better mimic like the intima and medial layers with smooth muscle and then endothelium. So I don't know exactly what type of bed we're creating yet, but I would say that our goal is to create those layered structures.
And I was just wondering whether it will be feasible to reconstruct something that indeed reflects vessels in distinct organs like heart and brain and so on. I mean, that of course is even further goal, yeah, another
Yeah.
layer, but whether
Well,
this is feasible at all. I'm
So maybe if I could dodge your question by saying, Rod was talking about meeting in Houston next year, maybe that's a problem for 3 or 4 meetings from now. But yeah, it's a really big challenge for us.
Okay,
Thanks.
thank you again. So I would like to move to the next speakers. Next speaker is Professor Ahmed I. Mahmoud from the University of Wisconsin-Madison. So the title is Dissecting the Sarcomere Metabolism Axis in Heart Regeneration. So please start.
All right. Well, it's a great pleasure to be here today, and thanks for the organizers, Jay and Kichi, for a great meeting. It's been a lot of fun and a great start for the day. So I'm going to talk today about some of our work with regard to metabolism and the interplay with sarcomeres for heart regeneration. And really our interest in metabolism stems from the fascinating roles that metabolism plays really as early as development. So metabolism and really the metabolic state of cells controls how cell fate determination takes place as stem cells differentiate. But really of great interest to us is the fact that early on during development, when cells are undergoing proliferation and growth, glycolysis is kind of the main metabolic state of these cells. Specifically, it's off-ramps as well, including the pentose phosphate pathway and the serine synthesis pathway. And basically, that is required for nucleotide biosynthesis, lipid synthesis, and so on. So pretty much creating all the building blocks that you would need for cell proliferation and growth. Now, as development progresses, the metabolic switch undergoes from glycolysis to mitochondrial metabolism, such as OXPHOS and fatty acid oxidation. Now, this sometimes happens at the end of development and sometimes postnatally, which is quite evident in the mammalian heart, where we know that within that first week after birth, the metabolic switch from glycolysis to fatty acid oxidation takes place. And from the heart perspective, it's a good thing that generates a lot more ATP and has a good functional role for the adult mammalian heart. However, the drawback is that results in loss of the cardiomyocyte cell cycle activity. It really is a main source for why postnatal myocytes start to lose that proliferative potential and subsequently the endogenous regenerative potential. So this metabolic state is not simply a correlation when it comes to cell proliferation and regeneration. In fact, a reverse metabolic switch takes place when regeneration happens in endogenous regeneration in response to injury. Now, this actually takes place in multiple cell types, including the activation of quiescent stem cell populations, such as neural stem cells, as they become activated and differentiate into neurons, and satellite cells in response to muscle injury as they generate new muscle. They really start switching their metabolism from oxidative phosphorylation back to glycolysis. But also remarkably, in endogenous regeneration of the zebrafish heart, where the border zone cardiomyocytes that undergo proliferation and pretty much regenerate the injured myocardium, they undergo a similar metabolic switch back towards glycolysis, really underscoring that conserved role of glycolysis in initiating that regenerative response. So it's quite an important way in thinking about how can we promote regeneration in a non-regenerating heart like the adult heart, and whether we can initiate such regenerative responses. So we initially were thinking whether we could approach that by specifically looking at succinate dehydrogenase. So succinate dehydrogenase was of interest to us because it's really the only enzyme that participates in the TCA cycle, as well as the electron transport chain. So it's complex 2 of the inner mitochondrial membrane. So it's a central mitochondrial enzyme. But more importantly, with respect to certain studies in cancer types such as pheochromocytoma, where mutations in succinate dehydrogenase has been shown to have a strong epigenetic effect and drives a metabolic reprogramming back to glycolysis, and that's what drives that cancer cell proliferation, which is simply known as the Warburg effect, of course. And as Thomas Brown nicely elucidated yesterday, and how that some accumulation of metabolites can indeed impact the methylation status by inhibiting the TET family of DNA modifying enzymes, as well as lysine demethylases. So we wanted to ask, well, whether inhibition of succinate dehydrogenase then, transiently at least in the adult heart, would it be able to kind of metabolically reprogram the heart and potentially activate a regenerative response. So when we treat the adult hearts or adult mice with a competitive inhibitor of succinate dehydrogenase malonate, pretty much daily injection for 2 weeks, and then we perform metabolomics to see how that impacts the metabolic state of the adult heart. And indeed, we start to see a shift from some TCA cycle metabolism towards glucose metabolism, indicating that that transient inhibition of SDH is sufficient to kind of promote that metabolic shift to glycolysis, which we were interested in. And now the question is that sufficient to reinitiate a regenerative response in the adult heart. So when we look after myocardial infarction, treat the mice for 2 weeks, and look at their hearts 2 weeks really after that last dose of treatment, whether there's an activation of cell cycle and cardiomyocyte turnover. And indeed, we can see a significant rise in the number of cardiomyocytes undergoing mitosis, as well as a significant increase in the cardiomyocytes undergoing cytokinesis as well, indicating that indeed SDH inhibition can promote adult cardiomyocyte cell cycle reentry. This is also evident in a significant increase in the number of mononucleated myocytes, which tend to be much smaller in size, and in contrast to a significant reduction in binucleated myocytes, these larger non-proliferating myocytes, underscoring that indeed cardiomyocytes turnover is taking place in these mice. But histologically, is that sufficient to regenerate the heart? Well, looking at that earlier kind of 2-week time point following our treatments, we do see a difference between the controls and the malonate-treated mice. We see a significant reduction in kind of that dilation that happens in controls. There's still some scarring, though, by that 14-week time point, so it's not fully regenerated yet. But 2 weeks later, which is after we stop our treatments, we now see a remarkable kind of restoration of myocardium in the hearts of these mice, and really just some minimal scarring around the ligation area. So again, that's a permanent ligation, underscoring indeed that this metabolic modulation can initiate an adult regenerative response. And again, we only also see by that 28-day time point that restoration of myocardial function as well, suggesting that that is not just making new tissue, but it's also a functional myocardium, which is really exciting. So we're very interested in exploring the mechanisms of this process and really how that metabolic modulation can initiate regeneration at both the cellular and molecular level. But one question that was really intriguing us is, we know that one major barrier for cardiomyocyte proliferation and cell cycle reentry is the fact that for a cardiomyocyte to divide, it needs to disassemble the sarcomeres. Really, it is a hallmark for myocytes to divide, where simply all the contractile proteins become kind of marginalized here at the periphery of a dividing myocyte. Here, that's positive for phosphohistone H3 as they're undergoing mitosis. So why would modulating that metabolic state have kind of overcome that significant barrier to cardiomyocyte division and subsequently to regeneration? So at the same time of during that metabolic switch that takes place early on in the postnatal heart, where a similar switch also takes place at the sarcomere level. And of course, many of you know are familiar with how the troponin I, the inhibitory subunit of the troponin complex, so it obviously plays an important role in muscle contraction as well as calcium sensitivity. But during that point, there's a switch from the slow skeletal troponin I to the cardiac troponin I, which is also a hallmark, as many of you know, of cardiomyocyte maturation. So again, this really takes place during that time point of metabolic transitions, which kind of suggesting that maybe there is a kind of an interplay between that metabolic and sarcomere switch that happens really at the postnatal heart and as cardiomyocytes start to lose their regenerative potential. So just to remind you a little bit about the differences between these proteins. So although they are encoded by two different genes, they're pretty much similar, except for an N-terminal extension that only exists in the cardiac troponin I. which has been shown to impact phosphorylation of the troponin I as well as calcium sensitivity. And when we quantify, again, these levels early on at P1, at the time point when myocytes can divide, the SSTNI is the predominant. At P8, as the cardiomyocytes start to lose that proliferative potential, it's kind of a 50-50 percentage, but within 28 days, the cardiac troponin I becomes the predominant isoform. And this is some top-down proteomic quantifications, which we've done with my colleague and collaborator, Inga. So what happens then if we think there's a correlation between that slow skeletal troponin I and the metabolic state? Well, we utilize the transgenic mouse line that overexpresses SSTNI and cardiomyocytes that was generated by John Solaro a long time ago. And we wanted first to know what the overexpression of SSTNI has an impact on cardiac troponin I. I mean, are they both expressed, or you have a combination of both, or SSTNI becomes the predominant one in the heart? So again, we went back to our collecting some time points from the transgenic mice at day 8 as well as day 28 and performed some of our top-down proteomics. And we do see that SSTNI is still the predominant isoform at P8. And interestingly, even by 28, there's only SSTNI and a complete lack of cardiac troponin I. It's kind of that the overexpression really blocks that cardiac troponin I, which is quite intriguing. We still don't know why that's the case, but we now have a model where we can really probe that whether that sustained expression of slow skeletal troponin I has kind of the reverse impact on the cardiac metabolic state and potentially the regenerative potential. So now we injured some of these transgenic mouse lines, and we did our injuries initially in day 7 old mice, so again to the time point when myocytes lose their proliferative ability and regenerative ability, and asked the question, well, compared to the controls, is there differences in their myocyte proliferative potential as well as their ability to regenerate? So at 7 days following injury, as we would expect from the control mice, there's really minimal mitosis going on. However, in the transgenic mice, there's a significant increase in the number of myocytes undergoing mitosis. This is also concomitant with a significant increase in cardiomyocyte cytokinesis as well, suggesting that just the overexpression of this isoform of the troponin I is sufficient to extend that window of cardiomyocyte proliferation. So 4 weeks or 3 to 4 weeks after the injury, we can also see a significant reduction in the cardiomyocyte cross-sectional area compared to the controls, indicating that that hypertrophic response that we would normally see in a non-regenerating heart is not really there, is also indicating that kind of the newly formed myocytes, which are usually smaller in size, seems to be happening a lot more in these transgenic mice. But remarkably, when we do trichrome staining and look at the histology of these mice, we see quite a remarkable restoration of the myocardium below the site of injury, in contrast to the controls, as you would expect, have these scars and thin walled ventricles, which really recapitulates that early neonatal regenerative response. So again, showing that activating or keeping that levels of troponin I beyond that window of regeneration is sufficient to maintain the regenerative state of the heart. But going back to our initial question of why we're interested in it, is that link between SSTNI and potentially the metabolic state of the heart. Well, what is distinct between overall proteomes between these transgenic mice and the controls? And we did perform some global proteomics as well in both controls and transgenic mice, but also comparing them to the normal regenerating heart. And as you can see, this is here the control wild type P7 non-regenerating heart. It's quite distinct from these two regenerative models. And by two models, I'm saying the normal wild type P1 neonatal injury model, as well as the day 7 injury model of these SSTNI transgenic mice. Even though they're quite similar, however, it still shows some distinct signatures between those endogenously regenerative P1 versus the transgenic SSTNI mice. And we look a little bit at the gene ontology of these genes, as we would expect from the normal P1 neonatal injury. There's quite an increase in the normal developmental processes, which we normally see, as we have shown in some of our early characterizations of the neonatal regenerative response. But what was unique about the transgenic SSTNI mice is that there's quite a significant change in their metabolic proteome based on their gene ontology. But when I take a bit closer look at some of these proteins, it involves components, regulatory subunits of AMPK, as well as an increase in some glycogenolysis proteins as well, indicating that there's a lot more breakdown of glycogen and potentially an increase in glucose metabolism as well. And there's an impact on some Wnt and TGF-beta signaling regulators as well. So, of course, again, it just shows that there is a remarkable impact on mediating that SSTNI expression in the postnatal heart and potentially a significant impact on their metabolic proteome. And of course, we're coupling that right now with some metabolomic and flux analysis to really probe the overall impact on the metabolic state of the hearts of these mice. But it still doesn't answer our question of how exactly does it do that. And just want to end up with one slide to highlight an ongoing collaboration with Daria Moshly-Rosen's lab at Stanford, who have some really interesting data that just came out recently on a preprint, where they show that some cardiac troponin I actually physically binds to ATP synthase, which is complex 5 of the inner mitochondrial membrane, pretty much the last step of ATP production. And they show that cardiac troponin I impacts ATPase activity, and interestingly, this seems to be abolished in SSTNI. And indeed, the predicted site of interaction of cardiac troponin I with ATP synthase seems to happen at the N-terminal extension, which is only present in the cardiac troponin I, but is absent in the SSTNI. So we think, or at least currently some of the ongoing work is to address whether that is partly how cardiac troponin I can impact the metabolism, and in contrast to SSTNI that seems to favor more glycolytic metabolism. So just to summarize what I've said so far is that we think, to kind of our approach and how we think about regenerating the adult mammalian heart, we believe that metabolic reprogramming holds a promise to kind of in line with all the other endogenous regenerative models, and might be a real evolutionarily conserved mechanism to kind of initiate regenerative programs. We think that specifically succinate dehydrogenase can be an important target to modulate that metabolic state, but there seems to be an interesting sarcomere metabolism axis that really plays an important role in mediating that metabolic state, but also in overcoming some of these barriers to cardiomyocyte proliferation and subsequently regeneration. So with that, I'd just like to thank the people who did the work, especially Timothy Abelo, who is a joint graduate student in my lab and Inga's lab, and where we get to combine some of our regeneration and metabolism work with Inga's elegant proteomics that you are all familiar with, and as well as thanking our collaborators, Daria and Ali at Stanford. And thank you for your time, and I'm happy to take any questions.
Thank you. Nice talk. Please start.
With great talk. I want to go back to a question I asked yesterday. It's fascinating that your mouse models don't develop hypertrophy. They just have this cardiac regeneration that you are describing only in response to injury. But the protein is there all the time. So why is this only happening when there is injury, but not during development or during regular, basal cardiac function?
Yeah, that's a great point. So, and we did, I didn't show the data, but we did proteomics without an injury in the SSTNI mice, and the proteome was the same. We only see that change in response to injury. I don't know why that's the case, but even it was puzzling to us. But,
but you see the same thing with your SDH
so with the
model.
well, with the SDH, it's different. So even in the uninjured, we do see the change in their metabolism. But because with the SDH inhibition, we do it transiently. So that's why we think, because we really stop our treatments 2 weeks later, we think that that metabolism shifts back. And that's why we see that improvement in function only almost 2 weeks after our treatment. So I think that maybe is the case with SDH because of our transient nature.
But I just want to bring up the possibility that you may not be seeing an increase in cardiomyocyte proliferation. It's just there is a different mechanism for protection against injury, which is independent of cardiomyocyte proliferation.
Again, I didn't show the data, but we've looked a few days right after the infarction, again, which is a permanent ligation, and we confirm that the infarct sizes are the same. There's cell death, there's no difference in apoptosis or the infarct sizes, but it's only we see that gradual progression of myocardial formation and a significant increase in myocyte turnover.
Okay, thanks.
Sure. Mauro.
Very, very nice story.
Thanks.
How do you envisage that you go from SSTNI to activation or proliferation? So there must be a mediator like YAP or the like that at a certain point gets activated. How do you envisage that?
Yeah, so, I mean, part of it, which is a little interesting to us, one part of it we initially thought, we still don't have evidence for it, is that it might be a kind of a structural thing in terms of just the differences between SSTNI and CTNI, but potentially it's the impact on metabolic regulation. We don't know why when we overexpress it also how CTNI is repressed. So there's clearly some sensing going on that kind of, as long as SSTNI is expressed, it's repressing cardiac troponin I.
Did you check if your proliferation is sensitive to YAP, so you can abolish it by inhibiting YAP?
A great question. We did send some of our slides to Jim Martin, so I'll ask him later.
Ahmed, very nice talk. I was very intrigued by the first part of your talk where, inhibiting SDH with malonate, making the heart go to more glucose, leads to this regeneration. There's data, which I talk about tomorrow, linking increased glucose utilization with adverse remodeling. So my question is, have you done any studies of duration of this treatment? In other words, is there a window where you see this, and then if you keep suppressing SDH, does it then become maladaptive over time?
Yeah, no, I don't think we don't have the data yet, but we've thought about it. One thing we are also utilizing now is that we have a fluxed mouse of SDH, so we kind of will have longer time. We can get a better insight of longer duration. We can also control a bit of mosaicism with our tamoxifen treatments as well. So we'll try to get a lot more insights in that, but we're thinking a little bit about it, but we don't have the answer to that yet.
Yeah, great story. Really enjoyed that. 2 questions. So one is, did you consider to also look in the cardiac troponin I mutants? I mean, they initially thrive, but then die because eventually also the slow isoform, skeletal slow isoform is inactivated later on. But initially, they are viable. So one might look whether this window of proliferation in these cardiac troponin I mutants is extended. So did you look at that? Or?
So cardiac troponin I mutants, which are like the actual the mature isoform, no, we haven't really looked at those.
It would be an opportunity. But second question, did you, another of these daring questions, did you look for histone modifications, which seems to me pretty obvious after malonate?
We are definitely, well, we're doing that after your talk yesterday, so definitely it's going, I already sent an email about it, so no worries.
Okay. Thank you.
Thanks.
Would be interesting to learn what you see.
Yeah, absolutely.
Let me know.
Great talk. I have 2 questions. One is, do you need both SDH inhibition and troponin I expression is required, or just one is enough?
So for SDH inhibition, that's usually good enough in our hands. We do looked at SSTNI expression in our SDH model. We don't see changes, but we think, again, because SSTNI is quite similar to CTNI, so maybe it's changing the phosphorylation of the N-terminal extension, but we haven't figured that part out yet. And potentially we want to do more proteomics, especially more kind of top-down and looking at the phosphorylation of the troponin I in the SDH inhibition. But usually SDH inhibition by itself is sufficient, but how it changes potentially phosphorylation of the troponin I is not yet clear.
Next question, does this phenomenon may occur in human cells?
We haven't done human cells yet. No.
Thank you.
Thank you. Okay, thank you.
Okay, so thank you for introduction. So anyway, I would like to say thanks to Professor Fukuda and Professor Zo for inviting me here. So it's my great pleasure to come here. So my title is Cell Sheet-Based Microcardiac Tissue Engineering. So we can fabricate such a beating cardiac tissue. So today I want to focus on some engineering technology. So anyway, this slide shows several technology for tissue and organ engineering. So most popular approach is 3D scaffold-based tissue engineering. So many researchers see the cells into such a biodegradable scaffolds. And also, some researchers using the mixture of gel and cells. So this is maybe feasible for the soft tissue. So many groups use such a concept. And also, some researchers use the decellularized tissue as a scaffold. Maybe it's not so good for the hard tissue, I think. So, and also, recently, bioprinting-based tissue engineering have rapidly appeared. So many researchers start to apply to the 3D printer to various types of tissue engineering. I don't know that it is, whether it is feasible or not for cardiac tissue. And also, some researchers also use cell fiber-based tissue engineering. But all these concepts use some material with cardiomyocytes or some cells. So on the other hand, as material-free tissue engineering, so our technology is one of the such technology, so cell sheet-based tissue engineering. And also recently, spheroid-based tissue engineering have started. So I'll explain briefly. So some spheroid or organoid was printed by machine to such a metal needle, or simply seeding to such a mesh mold. In both cases, by pulling out these metal device, so finally, a 3D tissue is fabricated. So anyway, there are many tissue engineering now. So today, of course, I want to focus on our technology, cell sheet-based tissue engineering. So maybe many researchers know this technology, but there are many younger people, maybe you don't know this technology I want to explain. So my boss, Professor Okano, have developed this technology. So cell sheet don't include any material, only two-dimensional cells. So by layering cell sheet, we can fabricate 3D tissue. So to get cell sheet, we have developed special culture dishes, temperature-responsive culture dishes. These culture surfaces are covalently grafted, very intelligent molecule, isopropylamide. These molecules are polymerized and covalently grafted culture surface, very thin, about 20 nanometers, so we cannot see. So anyway, this polymer surface changes their wettability depending on the temperature. So briefly, at normal culture temperature, 37 degrees, this culture surface is slightly hydrophobic. So the protein can adhere and cell adhesive. And on the other hand, by lowering temperature, this culture surface change to very hydrophilic. So protein cannot attach and cell cannot attach. So as you know, cells attach culture surface via several proteins, fibronectin or integrin between the cell and the surface. Usually we use the enzymatic digestion, typically a trypsinization. So in this case, all these proteins are destroyed, so cell can detach from culture surface. On the other hand, by using our temperature response culture surface, only lowering temperature, the connection between these proteins and the culture surface are released, so the cell can detach from a culture surface isolatedly. And these proteins on the culture surface are preserved, so cell can attach another surface easily. So if we culture the cells to conferency, a cell communicate to each other by cell-to-cell junction like this. So by lowering temperature, only the connection, these proteins underneath cell sheet and also culture polymer surface are released. So cells, conferent cells detach as intact cell sheet. This is our technology. So important point is this cell sheet don't include any material. This green indicate biological adhesive proteins. So anyway, we can get cell sheet. We have already applied this cell sheet technology to various types of tissue and organ engineering. And also we applied cell sheet transplantation to clinic in these 7 cases. Today, I have no time, so we don't mention the detail. So the lung, periodontal ligament, esophagus, cornea, heart, cartilage. This movie shows a simple transplantation. It's easy for the surgeon. So only put on the cell sheet on the damaged surface. So the cornea, this is a transplant fibroblast for pneumothorax case. So anyway, regarding heart failure, so we have started collaboration with Osaka University group, Professor Sawa and Professor Miyagawa. So they initiated clinical research. So first they use autologous myoblast sheets. So started clinical trial in 2007. And also, Thermo company started clinical trial 2012. And they got the conditional approval from the government to 2015. And also, Professor Sawa and Professor Miyagawa have also started the clinical transplantation of iPS-derived cardiomyocyte sheet. So they started in the 2020, so that maybe they transplanted 8 patients. So maybe, I don't know the detailed result, so I hope patient recovered from heart failure. Anyway, today I don't mention this cardio patch treatment. I want to focus on the myocardial tissue engineering. So our laboratory concentrated on the fabrication of the pulsatile, functional beating cardiac tissue. So I started 1999. In that time, I used a chicken cardiomyocyte or neonatal rat cardiomyocytes. And Professor Matsura in this room have started to using the embryonic stem cell derived cardiomyocyte first, and then he shifted to the usage of the iPS derived cardiomyocytes. So anyway, in our laboratory, we can expand the good beating cardiomyocyte using such a rotating bioreactor system. And this is a roadmap of our myocardial tissue engineering. So the big obstacle is the vascularization for scaling up. So many researchers perform the vascularization experiment. So our main strategy is like this. First, we prepare some vascular bed, including connectable arterial vein, and then we put on the layered cell sheet, including endothelial cells. If by layering and repeating this procedure, maybe we can make a thicker tissue. Of course, we can make a thicker tissue, 12 cell sheet in vitro and 30 cell sheet in vivo. So anyway, today I don't mention this vascularization technology. And also I have also studied such a microtube fabrication. Our final goal is a vascularized tubular structure. So anyway, today I want to focus on another topics, human myocardial tissue models. As you know, there are significant gaps between the normal cell culture and human body, and also between the animal model and the human body. So anyway, we can fabricate such a pulsatile functional iPS-derived cardiac tissue. So we apply this system to the several types of tissue model. So first is a planar case, and second is tubular structure, and third is a dome-shaped structure. Today I want to present these two, planar cell sheet model and also dome-shaped cardiac tissue model. So this demonstrate the contraction force measurement system of human cardiac cell sheet. First of all, we put on the human cardiac cell sheet onto such a fibrin gel-based device with such two grips. We hook these ends, both ends, to such a force measurement system. This is a load cell. So anyway, we can successfully measure the contraction force like this. So this slide shows increasing contraction force depending on layer number. So our technology, advantage of our technology, we can layer one by one, step by step. So this graph shows the contraction force, single layer, triple layer, 5 layer, 7 layer. So as you can see, the contraction force increased at least up to the 5 layer. However, in the 7 layer case, the contraction force decreased. So please watch this cross-sectional view. 1 layer, 2 layer, 3 layer, 5 layer, thickness increase. Also, 7 layer case, the thickness is biggest. However, as you can see, there are some delamination happened between the cell sheet. So it is caused by the oxygen or nutrition insufficiency cause such a maybe some necrosis happened. So the limit is 5 layer. We confirmed. However, honestly, single layer is enough for the analysis. So we usually put on the only single layer for the measurement. So these are several examples for the effect of the drugs. So the adrenaline, of course, increase the contraction force and beating rate. And also recently, cardio-oncology has been focused on. So we examined effects of several types anti-cancer drug, adrenomycin and epirubicin and bortezomib. So all 3 drugs significantly decrease the contraction force like this. And also, we examine the effect of drug delivery system. So as you know, so doxorubicin has the cardiotoxicity. So the doxil is a DDS of the doxorubicin. So the center core is the doxorubicin, and this layer is a lipid bilayer, and the surface is a very hydrophilic polyethylene glycol layer. So anyway, as you can see in the case of the doxil case, so we don't find any difference in the contraction force. So of course, doxorubicin decreases the contraction force. So and also the beating rate increase. So these data clearly shows the effectiveness of the drug delivery system. So all these data demonstrated that our system is feasible for the cardiotoxicity analysis. So this slide shows also the effects of fatty acid formulations in culture media. In that case, we add several fatty acids, like the uric acid, stearic acid, myristic acid, so on. So anyway, in the case of usage of such a mixture of fatty acid, the contraction force increase. And also we examine the oxygen consumption. So the result confirms the metabolic change happened from the anaerobic to the aerobic. So this is one case of the usage of this contraction model. So we have already published several papers using this contraction force measurement. So please refer our papers. So now we transfer this technique to the Nihon Koden company. So these guys are also PhD students in our lab. So anyway, they started to the drug toxicity testing service. And also we can use for the muscle tissue engineering, skeletal muscle tissue engineering. So we can make such a oriented human skeletal muscle tissue. I will show later the technology. So anyway, we can stimulate by electricity. So at the lower rate stimulation, so twitch happened. And if we stimulate at higher rate, so tetanus happened. It's the same as a normal living body. So anyway, now this system will be I hope this system will be commercially available in near future. So next topic is orientation of cardiac tissue. So we have performed two methods. So one method is such a technology. First, we make such a mold, V-shaped stripe mold. So anyway, we can fabricate it such a fibrin gel containing the V-shape pattern. So if we see the cardiomyocyte on the normal flat fibrin gel, of course, cardiomyocyte is randomly oriented. However, if we see it on this device, so cardiomyocyte aligned. So let's show the movie. So it's a flat on normal flat fibrin gel. So randomly oriented and randomly contraction. So next, this is aligned cardiomyocyte. As you can see, the contract from the left to right. So anyway, we can fabricate it such oriented cardiac tissue. So also we measure the contraction force like this. So significant improvement was observed in the case of using such a stripe patterned cardiac tissue. And also, Dr. Tagada in this room, so he performed this experiment. And also another technique is a surface modification. So base surface is a temperature response culture surface. And we patterned stripe like this with the hydrophilic polymer. And so fibroblast and muscle cell can align on this culture surface. But unluckily, so cardiomyocytes ignore this micropattern. So it's an obstacle. But I had an idea. So first we make such an aligned fibroblast. And then we put on the cardiac cells onto such aligned fibroblast cell sheet. In that case, cardiomyocytes start to align like this. This is a cardiac troponin staining. So green is all cells dimension staining. So the cardiomyocytes oriented according to the fibroblast orientation. And also we layered this aligned cardiac cell sheet vertically like this. So the vertical structure are preserved like this. If we layered several layers with little change the direction, layer by layer, maybe we can fabricate the in vivo-like tissue. As you know, in vivo tissue, such a cell orientation is change gradually. So anyway, this is one technology. So next, I want to mention about the dome-shaped cardiac tissue. So that in that case, we layer human cardiac cell sheet and fibroblast cell sheet, double layer. Anyway, by adding the culture media, so we can make such a dome-shaped cardiac tissue. We can control the dome size by adding the culture media. And also by OCT, optical coherent tomography, we can simultaneously measure such a movement. So anyway, we can calculate the area, so the volume. So the volume increases time dependently. So at 3 weeks, so they reach to the plateau. And also by adding the culture media, in the case of adding culture media, such a stroke improved to some extent. So maybe it is consistent with the Starling rules. So, but more pressure cause degrees of the contraction. And also we introduce the several sensors, of course pressure sensor, and we can control the inner pressure by using such a culture media reservoir. So anyway, we can control the inner pressure and we can realize isometric and isobolometric and isobaric mode. So anyway, in the isobolometric situation, we can measure such a pressure waveform. So if we increase basal pressure, so the peak, so this is stroke peak and also the max DT and minimum DT increase like this. So additionally, so we also add the valve system. So in that case, this is a cardiac tissue dome. So there are two valves, like in the human body. So we imitate the human circulatory system. So anyway, this is a valve. So we use a contact lens-like device. So open, close, open, close. So this is a flow. So significant flows observed. So of course, some backflow happened, but we confirmed the significant accumulation of flow volume. And also we can describe such a PV-like loop, but it's not consistent with the human body. Some such a not similar line observed. It may be caused some valve problem. I want to improve it. So, okay, so time is limited. I want to mention about something at a few slides. So anyway, we have started the cell sheet-based tissue engineering from 1990. So first object is, of course, realize the regenerative therapy. And next is tissue model fabrication. So we can get iPS cells derived cardiomyocytes. So recently, we started the cultured meat production. So of course, this is out of scope of this symposium, but I want to show you. So this is a layered bovine muscle cardiac cell sheet, 10 layer, no scaffold. So maybe it looks tasty, okay? So anyway, joking, but honestly saying, so I think it is very important for performing the various types of field research. So in our laboratory, so many researchers gathered, they perform the regenerative therapy, tissue model, and cultured meat. So basic technology are common to all applications. Of course, final focus is different. So in regenerative therapy case, we must think about safety and efficacy. Tissue model case, maybe the similarity and reproducibility. And regarding culture meat case, it's cost and taste. So differ. But I think it is important to communicate among the researchers who are engaged in each research field. So anyway, this is my future challenge in molecular tissue engineering. So one main is sterilization, as you know, and maturation. So there are several ideas like this, but there are many things to do and to be solved. Last night I mentioned, so please, I ask younger people to join in such a bioengineering research for cardiac tissue engineering. So final goal is rescuing many patients. So anyway, I would like to say thanks to all my colleagues, and some part is granted for government. Thank you for your attention.